All right, so we're going to continue talking about our mutations. Our last one here is going to be what's known as a frameshift. Because what happens with a frameshift mutation, you have one of two things that happens, all right? Either you are inserting nucleotides into the gene, or you are deleting nucleotides from our gene, okay? And what that's going to do, if we put up a sequence here, all right, and that's just random sequence, okay? So this would be what we would call normal, okay? And you see our codons are going to go AUG, CCC, AAT, TTC, or TCC, GGT, okay? If we have an insertion, we're going to still start out AUG. Oops, I wanted to put the insertion in a different color so you see it better. So say we end up inserting two G's here because maybe there's a little bit of stutter in the DNA replication and we get 3 guanines put in. A, T, T, C, C, G, G, T. What that's going to do, we've got an AUG, that's going to be the same. But now our second amino acid, instead of being CCC, is GGC. All right? And what we've done is we've shifted our reading frame. All right? And that shift in the reading frame means that different proteins are going to be inserted. All right? And that's a bigger change than what we had before. If we do a deletion, okay, we may have our AUG, let's do CCC, and then we go TT, CC, GGT, okay? And we end up losing those 2 nucleotides there. Somehow they get cut out. All right, we've still got the AUG, we've still got the CCC. Now what we've done is we've shifted, and our third codon, instead of being AAT, is TTC, okay? Then we go CGG, and so on, okay? So really what we're talking about with frame shifts, okay, we're talking about changing how the ribosome reads our nucleotides. Okay. Now keep in mind here, I put up the DNA sequence because what has to mutate is the DNA sequence. What will happen and change in translation is the RNA that's made from this DNA template will be different than the RNA made from this DNA template or this DNA template, okay? And that's what gives you the different protein, okay? Again, you can have something happen where you end up getting a protein cut off and made too short. You can have proteins, if that frameshift mutation changes things so where you've normally got a stop codon, now that stop codon is read differently, so it's read and an amino acid is inserted. Because it's no longer a UGA, it becomes a CUG. That's going to insert an amino acid, and that stop codon won't be recognized. The ribosome is going to keep inserting amino acids until it reaches the next stop codon in the sequence. Okay? So frame shifts tend to be a little bit bigger. I showed the insertion and the deletion here as 2 nucleotides. It could be 1 nucleotide inserted or deleted. It could be 20 nucleotides inserted or deleted, okay? So there's a lot more change in that. That kind of falls into the bigger rearrangements that happen. Although certainly frameshift mutations can also be a type of point mutation, as long as you're talking about only 1 nucleotide being inserted or 1 nucleotide being deleted, okay? So that's our frameshift mutation. When we're talking about mutations, what I want you to keep in mind, all right, what generates mutations are substances known as mutagens, all right? And our 2 main mutagens that are going to be out there, radiation, whether it be ionizing radiation or UV radiation, that can result in changes in the DNA sequence. Chemicals also are often the sources of mutations, okay, or mutagens. So they cause changes to the DNA sequence, okay? So keep that in mind. If you have any questions about mutations, please let me know.